Uh, Good evening, participants. Am I audible to everyone? Yeah, am I audible? Anyone can interact? Yes, yes, yes. I think it's audible for you. Just checking my audibility to the sound. Okay, can we discuss the last session topics? So, what exactly did we discuss in the last session? I think we have seen enormous content in the last session, isn't it? Today I slow down. Okay, I take you a few concepts which I can make you understand. Systems biology. So overall, we discussed about system biology. So what exactly we discuss in the system biology? What all come under system biology? So we have discussed various what science subjects. You remember? We discussed various very good. We discussed various omics, isn't it? So one is genomics. Other than genomics, what else can we give as an example? Yes, we also discussed about system biology markup language. Okay, so if you want to practice any part of omics, genomics, proteomics, interactomics, transcriptomics, metabolomics, for all this, we can able to build the components. That is, for example, if you want to build protein-protein interaction networks, to build protein PPAs, you need a system biology markup language. Got it. Not only for protein-protein interactions, also for gene-gene interactions, gene regulation network interactions. For all this, you not only need a general concepts like graph theory, statistics, etc. Of course, if only if you have a language, then only you can develop the software components. The best example I also provided is that you can apply the same SPML in studying about compartment models. Okay, that is if you're working on disease models. Okay, that's what we call it as a bio models, which we use SPML for development. Okay, so if you go for disease models, especially pharmacokinetic compartment models, so two compartment to multi compartment, like whatever drug you feed, whether it is reaching a desired target or not, and how much is going to unutilized, and how much is excreted, all this information we study in the form of kinetics. Okay, so the rate of reaction kinetics, okay, biological reaction engineering, this is what we call it. So you are linking so many subjects in this systems biology. And whatever I am telling system, it is a biological system. For example, if you are studying about human diseases, human is the system here. And if you are studying about production of proteins, recombinant proteins, okay, through genetic engineering techniques or through some normal bioprocess engineering techniques, then what bacterial cell, what E cell used for development of the humanized insulin? Okay, so that's a pro-insulin. So that's what pro-insulin or growth hormones or whatever proteins, the interferons, GM-CSF, granulocyte macrophage colony stimulating factor. So these are the most widely produced proteins from the bacterial sources. So if you study about the bacterial cell, you're not going to study the entire thing. You study cell by cell. Even if you're studying about heart, cardiovascular diseases, you study only one heart cell at a time. That you take it as a unit, and then you study. Just like how you see in neural networks, a neuron is a unit. Okay, so we can able to interlink with how many units we want, and we can study detail in system biology markup language. Okay, give me some examples which we have covered in system biology software. I need some softwares. So like each and every software is for unique application, isn't it? I have provided you demos on yesterday with system biology softwares. What softwares I have covered? LADMET, that is liberation, absorption, distribution, metabolism, excretion, toxicity. That is in pharmacokinetics. If you want to determine this LADMET properties, okay, you can use such a kind of, actually, that is What exactly we use actually? We use Swiss ADME. If you want to determine the ADME characteristics, you can go for Swiss ADME. But this is not pertaining to system biology, but this is something. So, okay, we can link. I can't tell it's fully not linked. You're right. We can able to link because pharmacokinetic, of course, link with the pharmacodynamics. Okay. So, but you are going to study about, if you want to specifically study about the system, then I can give you an example like very good, MATLAB. This is what I anticipated. We have SimBiology tool. Okay. We have specifically called SimBiology tool, system biology tool. Okay, which we can use it for working in various applications, especially studying the drug release during drug protein binding in compartment models. So all this information you can able to find in SimBiology. We can simulate it, we can run, and we can check the bioequivalence, bioavailability, etc. in the MATLAB SimBiology tool. Okay, then what else did we discuss other than MATLAB SimBiology? We have discussed PPI, isn't it? I showed you an online software. Do you remember what software I showed you? I've showed you a software. I better close the WhatsApp. Yeah. So what is the software I used? You forgot? Can I take you to that software once again? Hey, then you have forgot. Then I'll be taking you. Yeah. So I take you to the software in String DB. You remember? String DB is to study about protein-protein interaction networks. So here in this string DB, okay, again I show you the home page. If you go to the home page, oh, directly taking you to this page. Okay, if you click here, still it's there. Okay, we came to the next page. If you type simply stringdb.org, you can get how many number of protein-protein interactions are available. Around 23 million protein interactions are available, and nearly 1,500 species information are available. Okay. Is my video and audio quality is good? Yes or no? Just type that because I want to check because on yesterday I got a feedback from a participant that video quality is low. So is video quality low or okay? Yes or no? Good. Okay, thank you. Now let me take you in string because if I take you to the protein-protein interaction networks, even the internet, uh, uh, it will delay that data what you're going to load from the internet. It delays. So I'm checking how. I'm going to type a kind of a protein at present. So I'm going to type a protein called EGFR, epidermal growth factor receptor. Epidermal growth factor receptor. Simply, I'm going to search for that kind of a receptor. Defaultly, it takes you to the Homo sapiens protein, but I also got nearly 574 proteins for different species. Okay, so if you want to go to the Homo sapiens, simply click continue. You get a kind of proteins linked with this hub protein. Each of our is our hub protein. These are the interacting proteins. If you want to know more information about the interacting proteins, just click here. You find ERPB2. What is ERPB2? It is a receptor tyrosine protein kinase ERPB2. Okay, you get some information on that. You get also the motif information and whatever information you get, okay. Even you can link with the UniProt and you can find the information of that. So if you want to increase the number of protein interactions with epidermal growth factor receptor, then in settings, you can go inside and instead of 10 interactors, you can give the custom value as 100. Now I give a custom value as 100. Okay, and now simply I'm going to click update. If I click update, obviously I can find 100 base protein interactions with the epidermal growth factor receptor. This is our hub protein. So this protein you can find so many interactions. And if you want to know how exact the protein-protein interactions, gene interactions, all these things comes under system biology. It's a product of system biology. Like if you want to specifically uh, uh, target this gene, that is, if a person is suffering from a specific type of cancer and the EGFR is the major gene, okay, then you can't simply develop a drug and treat a drug by targeting this receptor alone. One should also try to target the interacting proteins. Even though if you get 100 proteins, what is the most interacting protein? That is, for which is going to more upregulating or downregulating the proteins. Okay, all these things, if you try to study, of course, is a part of a system biology. Okay, so for this, if you want to know about how to go for analysis, okay, so before going for analysis, I can show what are the various options available here. So if you will see the viewers, okay, yeah, if you want to see the networks, okay, the types of networks you can find from here, okay, or if you want to see the form of experiments, what they have done to develop these interactions, protein-protein interactions, you click the following experiments. So you can find in lab experiments according to anti-tag co-immunoprecipitation, okay, assay. So this is an assay they have used. Immunotechnology is utilized. Okay, so we can see so many techniques. One is anti-tag co-immunoprecipitation. The other is proximity-dependent biotin identification assay, and we can see antibody co-immunoprecipitation. The anti-tag is different, antibody is different, tandem affinity purification. So in each and every wet lab techniques, you can find some series of proteins are connected in the network. In this case, if you have nearly some 14, 15 proteins, 15 plus 8, 9 proteins are there according to this assay. In another bioassay experiment, you find 241 plus 15 more proteins are there in the network. Okay. Do you remember? Initially, when I just opened that EGFR, I got only 10 protein interactions, best 10 protein interactions with EGFR. But according to this experiment alone, this is one experiment done by a scientist. Okay, so you can find around 260, more than 260 interactions out there. Okay, so we can also customize value up to 260 if 260 is the maximum. Here, if you see, I got nearly 567 plus 13, around 580 interactions are there. So I can take it maximum to the 580. I published few papers on system biology in reputed journals like Scientific Reports, Informatics in Medicine Unlocked, etc. So in that, I have tried with 1,000 proteins, 1,000 interactions. From 1,000 interactions, I screened top 100 upregulating and downregulating genes based on p-value. So how we can able to screen the interactions where, on regulation, upregulation, downregulation. For this also I can use MATLAB. And of course, Cytoscape is the best analyzer to analyze the protein-protein interaction networks. Okay, so you can link the Cytoscape visualizer. So if I ask you the software, so you can tell the first software you can give is MATLAB for symbiology and Cytoscape for visualizing all the type of network interactions, protein-protein, gene-gene network, gene-gene interaction network, so metabolic networks. Okay, so DNA protein interaction networks. So for all this, you need a Cytoscape visualizer. Okay, that is a second important software. And third, of course, the online software like String is the largest database for protein-protein interactions. Okay, so this database is obviously linked with UniProt and most popular databases. If you want to get it interaction databases, even you can get information from UniProt. Okay, so this is something about how we can find the information, and these are the wet lab techniques, and we can start from 1,000 interactions, and we can screen based on our customization how we want to use an advanced filter for uh, filtering the for knowing the most important proteins. So experiments and databases. So databases we link with metabolic pathways, protein complexes, okay, other kind of protein-protein interaction networks. So we have plenty of databases and tools linked with it. Okay, of course, even through the String database, you can directly take your data to the Cytoscape visualizer. Okay, so this is database knowledge. We have annotated pathway cake that is you can link with the 466 plus nearly 30 proteins, nearly 496 proteins, or linked or in this cake pathway. So not only can see the network of proteins, but also in the pathways and what kind of diseases, all this information, you can have an idea on that. And not only that, you can see multiple kind of annotations are given. Okay, this is one such a database entry, and this is a second database entry. This is related. The major title of the cake entry is Pathways in Cancer. Pathways in cancer, around 496 proteins are linked with the EGFR. And there is another title, the cake title, that is EGFR tyrosine kinase inhibitor resistance. For this, you have this much, less than 100 proteins are there. Similarly, even you can find EGFR is linked with so many interlinking pathways like phosphoinositol 3 kinase, AKT, and ERPV. So many pathways information. It takes you to the pathway, and you can find how the proteins are interacted. So you can find much information. Okay, you can find about concurrence, co-expression, fusion of genes, neighborhood information. Okay, all this in this stringdb.org. So if you want to download that interactions, whatever I showed earlier, that 100 interactions, these 100 interactions we can download in the form of Excel sheet. You can download in the form of .tsv file. TSV. It means tabulated separate value file. The tab separated value files, you can find this data. You can open in Excel sheet for editing, and you can use the Cytoscape visualizer for seeing the interactions. Okay, let me join once again. Yeah, gene fusions. So, so you can this database is taking some time to load. Okay, meanwhile, I can take you to the next database. The next database, what I'm going to show is gene regulation. You have protein DNA binding, isn't it? So generegulation.org, protein-DNA binding. Okay, so under that, you can find different softwares. That is, if you're working on DNA protein binding, you can use this software. We have also databases. Okay, so many data sets are deposited in this. So if you are working on that DNA protein interaction databases, you can find the information from this website URL. Okay. So I just copy the paste here. Can you provide the papers on system biology? Sure, I can provide you. Uh, I can provide you in the form of DY. Okay. So you want it now? So the first article is published in Scientific Reports. This is from Informatics in Medicine Unlocked. So all are commercial. Uh, so both the publication fees are more than 2 lakhs. So this is Informatics in Medicine Unlocked. Okay, so these two papers are pertaining to system biology approach. One can leisurely find the information when you find free. Okay, now let me take you. I was explaining you about gene fusions information in the string. Okay, so like the conservation domains. Okay, what gene is conserved and what gene we can fuse. So related to that information, you can find from here. Okay. Uh, And I was speaking about the protein DNA interactions. You can go to this URL. Okay. And for the protein DNA interactome. Okay. So this is a database developed, this is a tool developed by John Hopkins Hospital. Okay. So John Hopkins Hospital has developed human protein DNA interactome. Human DNA proteome interaction, isn't it? So protein DNA intacto. Now I close it. So simply you have to paste the FASTA format of a DNA sequence, and the default Homeo box is there. If you know any specific domain specific, you select the domain and you can click submit. Okay. So one can use the FASTA format as the input. Now I just click enter. This is human DNA proteome interaction, and this is for DP interactor. Okay, so that is if you want to find information of different motif proteins, you can try with this. Okay, DP interactor, database interactions. Okay, DNA binding proteins uh, information can also be found from here. The DNA. So till now, whatever URLs I'm pasting is DNA binding proteins, and myself, I am working in protein bioinformatics lab in IIT Madras. So PBI lab in IIT Madras. So he is my mentor, Professor M. Michael Gromiha. He is India's #1 bioinformatician. So his specialization involves in development of databases and software tools. So if you want to know what exact, he has developed maximum number of tools and databases. So whatever I have showed you, something like DNA protein interactions, you also have RNA protein interactions, carbohydrate protein interactions, so many lipid protein interactions. All the kind of tools and databases are developed by him and his his other research scholars. I mean, his research scholar. Now I can show you. So this I can find from resources. Okay, so if I click the databases, now you can see various databases are shown. So this is a database for checking binding affinity and neutralization profiles of coronavirus related antibodies. So and the MPID is a database for binding affinity of membrane protein complexes and their mutants. So this is the website, and this is the URL. So if you to go to this URL, you can practice, you can try with your inputs. And this is RNA small molecule interaction miner, a database for binding affinities of protein nucleic acid complexes. So ProThermDB, many people are trying to use this. Okay, this is uh, even you can try this if you are in this kind of a research. Likewise, you see plenty of databases are developed by him. Okay, by him and his team. And now take you to the tools and link to the publication. So he is having both the tools, so link to the server. Okay, if you want to interact with the tools, okay, like PDA, thread, you can click here. To go to the publication, go to the, you have to click the left. Now this is the software. Okay, so click here to start prediction. Now see the inputs. Okay, so feed the PDB ID and whatever inputs we have to give, and we can check the protein DNA binding affinity, how whether a DNA is going to bind with the protein or not in DNA binding proteins. So this information we can predict from this tool. So actually here you can find a link for the tools. Here this is a link for the publication. Okay. So if you have excellent computing facilities, even myself, from the grant, I am working a simulation. We purchased a simulate server for installation of simulation software like Chromax, and I'm working on it. Okay, so these are some of the tools. Okay, especially for all these tools are related to systems biology. Okay, so to know more about him and the tools, etc. Okay, so now I can take you to the synthetic biology. Okay, so till now, whatever I explained is related to system biology. I can also show you one more interesting software. called FunCook because if you want to practice, if you want to train on mini projects, okay, this knowledge is essential. So instead of just training you in the theory even you can learn from Wikipedia or even artificial intelligence tools like ChatGPT. But how about the practical hands-on experience? So you're registered one day to gain more knowledge on this, isn't it? So I show you something in online tools, how we can able to work. Okay, so now I'm going to type the same EGFR in FunCook database. You know, even this is a protein-protein interaction database. So if I just click search, okay, see the output. The outputs are somewhat different when compared to this. Okay, if you want to see the networks, simply go to the network. I just distinguish how you find. Yeah, so the networks are like this here, isn't it? It's getting loaded. Now see here, the output in FunCook. This is output in FunCook. So you're getting so many kind of interacting proteins. For example, if I'm working on breast cancer, okay, so I'm going to click breast. So only few proteins are shown. The remaining nodes are hidden. If I'm not selecting breast, all tissues, you find more proteins. If I click breast, even EGFR got disappeared. So EGFR is not the major target receptor. So that's what you can understand. Okay. And after that, okay, even if you want to filter it through the pathway, for example, if you want to know how this EGFR, how you can link the network of proteins of breast cancer with MAPK, I just selecting this. This is one filter, filtering by tissue. This is filtering by pathway. If I filter by pathway, you got these proteins, totally 5 proteins. Okay. So so you also have an identifier. If you click this, it takes you to the EMBL website. Okay, so so likewise, you can able to see the protein-protein interactions, and you can filter accordingly. If you want to download this network in .tsv, that is, if you want to edit the interactions, the distance of interactions, you can make it out. Okay, using this x .tsv, which we can open it in Excel. And if you want to import the same in the form of portable graphics format, and that is in the form of image, you can download from here. Okay, you got it. So here, even here, if you want to export, that is, if you want to download this protein-protein interaction, click this tabular text for output TSV. You have to click this download. Okay, either click this or I think this is the best one. Short tabular text output is the best one because it can be opened in Excel and Cytoscape. Okay, so either you can open in Excel or you can open. If you have installed a recent version of Cytoscape, okay, if you click send to network to escape, directly it opens in the Cytoscape visualizer. Okay, so this is something about systems biology, protein-protein interaction, DNA-protein interaction, even RNA-protein interactions. So I told you I have given a hyperlink also, isn't it? If you go to the hyperlink of IATM, you can find all this information. Now, let me take you to the synthetic biology. So, what is the difference between synthetic biology and system biology? I told you system biology is majorly you study about the integration of omics, and you obviously have to study the network theory if you work on network biology. Okay, so network theory, statistics, and biology, isn't it? So, here in a synthetic biology, there is an interdisciplinary branch of biology and engineering. Okay, the engineering mostly we discuss on genetic engineering. Okay, so if you see this subject, there's a combination of biotech, genetic engineering, molecular biology, molecular engineering, system biology. Also, we are integrating system biology, but majorly you study the major biology, all the types of biology with genetic engineering. Okay, of course, computer engineering is also involved because, however, you find the building of components. You remember I told you how to use MATLAB and how to go for building the models and analyzing. Similarly, here also we have to build the models. So, if you want to build the models, one should have even a basic knowledge of computer engineering, control engineering, okay, and the biology. Yeah, of course, you should also have a knowledge on evolutionary biology because this synthetic biology is majorly used for production of recombinant proteins from genetic engineering through microorganisms. That is the major application, that is the major use of synthetic biology. But system biology is majorly used for precise targeting of diseases. If you want to precisely target any disease, specifically cancer, okay, then you better go for system biology, where you can take even genome. Genome sequencing is, of course, a major uh, chapter to study. Okay, so we understand. So you're for creating genetically engineered circuits. Okay, like or also integrating something relevant to trivially. You heard about how we can find a circuit diagram. Similarly, in genetic engineering, we can link the artificial components linked in the genetic circuits. So, what is it exactly? Artificial biological system for research. Here you have biological entities like enzymes, uh, genetic circuits, and cells for the design of existing biological systems. This is what you find in synthetic biology. If you want to work on synthetic biology, one has to learn on synthetic biology open language. For system biology, it is system biology markup language. But for synthetic biology, it is open language, SPOL. Okay. And this is the URL, SPOL standard.org. And if you see what exactly you find in the software, in the synthetic biology open language, you find these symbols. Promoter, so for a promoter, this is the standard symbol. For complementary determined site, you find like this. For like how you find parking rules. If you want to go for learn for getting a driving license for a two-wheeler or four-wheeler, so for each and every instruction or warning, they provide a symbol, isn't it? Similarly here, for synthetic biology circuit, that is, if you want to uh, uh, create, for example, if you take a tomato, okay, in a tomato that is genetically engineered tomato, especially compare a raw tomato with Bengaluru tomato. Here we call it as a Bengaluru tomato. The Bengaluru tomato is very fresh, and the skin thickness is thin. Okay, the Bengaluru tomato, the thickness of tomato skin is thin, and you can see the flesh is more. You find less seeds. So these are some of the uh, characters we expect from this kind of a vegetable, tomato. Isn't a tomato, fruit or vegetable, whatever you consider. So generally we expect a thin skin, and the color should be red color. It should be even. It should not like green, orange, something, three different colors, three or four different colors. We expect a good color, nice color, like a red color, an even color. We expect even flesh, and we don't expect seeds. So for each and every parameter for a trait, okay, you have to go for gene silencing. So if and if we go for gene silencing, okay, like silencing production of seeds or production of maximum seeds, silencing the uh, thickness of the skin, okay, the tomato skin. So all these things will give you a kind of a better genetically modified tomato. Similarly, think about now the genetically produced guavas. Nowadays, we are getting very large covers, isn't it? So you can find lot of differences in the phenotype characteristics, isn't it? So for all these things, okay, if you want to engineer 4 to 5 different genes, and if you want to have all the 5 engineered genes, okay, so you can have all the engineering genes in a circuit. So you are studying whatever you study in the computational model, they are trying it in wet lab, and they are obviously producing that kind of seeds, and obviously from the seeds they are producing that kind of fruits. Okay, so the promoter, CDS, ribosome entry, terminator. So not I am telling you that these kind of a synthetic biology concept can also be used in plant biotechnology, not restricted up to animal biotechnology. So instead of sacrificing porcine, swine for insulin, okay, we can take the human insulin itself, and we can go for genetically engineered with E. coli, isn't it? So one should try with this kind of synthetic biology concepts. Okay, so now we got some of the terms, okay, some of the components used in building the genetic circuit. Okay, now let me take you to the synthetic biology. A biological part is a sequence of DNA that encode for a biological function. For example, promoters or protein coding sequences. So generally, you link multiple components, okay, to form a genetic circuits. Okay, this is a part of the genetic circuit. Did you hear something about artificial chromosomes, bacterial artificial chromosomes, yeast artificial chromosomes? How these are constructed? How a vector is constructed from the plasmid? So one should have a knowledge on synthetic biology for these. Okay, the artificial chromosomes construction, and even whatever I mentioned. So okay, so for all these things you should need a synthetic biology. You have to link different components, okay, promoter, inducer, and other parts of the gene, okay, and then you try to form a circuit, and whatever you want you keep it, and whatever you want to silence, you can silence the gene. Okay, so this is some of the information. Okay, like even if you want to know about promoters, so what for promoter is used, some basic terminologies. Okay, you can find in the synthetic biology website. Okay, I showed you that URL, isn't it, earlier? You can go to the URL and get this information. So what is the function of ribosome? If you are very new, you're learning from the scratch, okay, then you can go with that website URL and learn each and every component present, the technology is present. Okay, in overall, if you see the illustration of the construct engineered therapeutic microbes based synthetic biology approach, we can see some examples. Genetic part development. Okay, so like so in next generation sequencing, whatever you sequence, okay, like for example, if you want to identify the species, what exact technique we use? 16 years RNA sequencing, isn't it? To identify the species. Even if you want to identify a mummy, okay, through carbon dating experiments, one should also have to go to the sequencing experiments, okay, to know the age of a person, a dead corpse, if you want to find. So then one should go for a sequencing, and then they can come up with a, of course, sequencing linked with carbon dating gives you a kind of a best knowledge on that. Okay, so likewise, we can also go with the genetic circuit construction. So whatever concepts we find in triple E, that is logic gates, AND gate, OR gate, NOR gate. Okay, so switching off one circuit, switching off one gene to the next gene. Okay, if one gene is getting silenced, it's going to move to the second gene in the circuit. Then obviously, what is going to happen? What kind of product is going to come? So all these things we can able to study using this. This is a kind of a genetic circuit diagram, example of genetic circuit. Oh no. So this is a computational design of genetics, how you find. And this also is for testing platform. Like if you want to check with the organoid, okay. Like for example, if I'm working on liver cancer, so in liver cancer, a liver is an organ, but in a liver, you find so many organoids. Okay, you find nearly 6 to 7 types of major cells in liver, 6 to 7 major cells in liver. Okay, and whatever I'm mentioning you that maximum of 7 cells. Okay, even you have a different organoids. In one organoid, you find some 4 cells. In another organoid, you find a different combination of 6 cells. In another organoid, you find 7 cells. Okay, so even you can test whatever you can able to see in the organoid, you can able to construct in the computation, you can check it out and even cut on a chip. So whatever you can try with in vivo experiments or testing experiment with human, we can able to make the kind of I mean we can work similarly even through the chip. So we can have in the chip, we can have the kind of a cells, okay, an array of cells, and we can try with it. Okay, if you are working with uh, any genetic engineering principles. So likewise, we have a different applications. I told you it can be used in plant biotechnology, animal biotechnology, microbial biotechnology. Okay, in all the areas it can be able to find the significant. Now let us go to this topic called microarrays. Microarrays actually, if you see, a microarray looks like this. An instrument looks like this. Okay, and you have a kind of nearly a robot to print around 10,000 spots. If you want to fill a very small sample, okay, then you can go for using microarray techniques. You know, microarray generally is for measuring gene expression for diseases like cancer. So, if a patient is suffering from blood cancer, the sample tissue take collection from the blood is easier, isn't it? Because a 75 kg male or female is going to have average of 6 liters in her body. So it's easy to take a sample and we can able to diagnose it easily. But if a patient is suffering from pancreatic cancer, what is going to happen? You have to go for biopsy of pancreas. If only if you dissect pancreas from human, then only we can correctly diagnose. You know the pancreas size? The pancreas is very less in size, isn't it? In centimeters. So if you have that much small pancreas, if you dissect it, and how many days it take after biopsy, how many days it takes for regeneration? It may take in years. So meanwhile, the patient is going to get weakened, and there are more chances of fatality. So instead of taking a large sample from the pancreatic tissues, okay, we can take a small sample, like even 10 microgram or 20 microgram is sufficient. 20 microgram of tissue, we can able to make it up to 20 ml, that is around 20 microgram and 20 ml. Whatever sample you take, okay, that sample you can able to, if you feed it to the microarray robot machine, it can fill up to 10,000 to 20,000 spots. Is that possible with the pipette man? No, it's not possible with pipette man. Zero point 0.1 microliter is the least sample which we can able to pipette. But I am telling you the 20 microliter sample, you can fill it up to 20,000 spots in nearly 1.5 centimeter, 1.5 centimeter chip. This looks like a glass slide. Okay, you just have a kind of a small area, nearly 1.5 to 1.5, where you can fill this much spots. So the diagnosis will be made more easier if you take all these 20,000 data together. Instead of taking a single data, you are going to take 20,000 data. Okay, so you will be precisely can diagnose. Like even after giving the drug treatment, if you want to know how what is the gene expression after the drug treatment, okay, like after a month, what is the gene expression? After 2 months, what is the gene expression? Then one can able to go for microarrays to estimate the gene expression. Okay, now what are the steps in microarrays gene expression analysis? How it link with bioinformatics? You know, in pharmacogenomics data management, okay, the major technique what we use, okay, is the microarrays under bioinformatics analysis. So under bioinformatics database management, microanalysis is the major technique which we use in pharmacogenomics data management. Like what do you mean by pharmacogenomics? Like giving a drug and checking how you find the genes expression in the genomes. Okay, that's most important, isn't it? Like for a disease, diagnosis of disease like cancer. So if you see, I think this. Yeah, this is the best slide which can explain you the steps of DNA microarray. In microarray, defaultly we call it as a DNA microarray, but we also have protein microarrays. So what is DNA microarray? How the sample is taken, how it is analyzed. So generally a sample, whatever you take, okay, we take the healthy, suppose we take is sample one, and diseased if you take like a sample two. Okay, so We generally going to take RNA as the sample from the cells. Whatever RNA you take from the cells, mRNA, we are going to convert it as cDNA through reverse transcription techniques. And after converting RNA to cDNA, we are going to label this RNA through fluorescent dyes like CY3 green and CY5 red. So if you label with these kind of CY3 and CY5, okay, then obviously you find two different cDNA or tagged with the probes, fluorescent probes. So fluorescent dyes. Okay, so whatever you had, you take together and you mince together. Okay, and we go for hybridization protocol. In hybridization, this RNA can also mingle with this RNA through hybridized. Okay, so you get a kind of a hybridized strands. Okay, but if you want to go for analysis, okay, generally you pass a red laser for detecting the red fluorescence and the green laser for detecting the green fluorescence. So green laser gives you the green fluorescent images, okay, in the microarray. And the red laser is going to emit only the red fluorescence images. So both the images you have to superimpose. If you superimpose these true images, if you find green and red, okay, obviously if you mix green and red color, what color you get? Yellow color. So here you find the first spot 0, comma 0, you can find green. Here also 0, comma 0, you can find red. So you get like yellow color. But here the dark green you find, but here you find red, but here you find red color. It means that diseased expression is poor when compared to the healthy cell expression. If green is considered as healthy, so just even if you see from this microarray, if you superimpose it, you find many spots in green color. So I can tell if the patient is given a sample for a diagnosis of cancer, I can tell around 20 to 30% expression is there. So based on the intensity is based on the number of green colors and based on the intensity of green colors and based on the same whatever I mentioned for yellow colors and red colors. The yellow color is the combination. Okay, if you get a equal green and equal red, then you get an yellow color, a bright yellow color. If uh, if you get a kind of a light yellow color, very light yellow color, then you find more disease gene expression in that spot. So whatever colors you get, light or dark, from green, red, yellow, and the black, what is if you have any black spots that are unfilled spots, that is the sample is not filled in that spot, then you won't get any proper kind of green or red color fluorescence inside. So even if you superimpose, you get just a black color is retained. Okay, so you get these kind of a colors, and through the statistical analysis, if you have 20,000 spots, so you are going to estimate it through parallelization, your normalization procedures. Okay, so whatever we can able to learn the gene expression through image acquisition and analysis, okay, that gives you a better kind of a result, and it directly tells you how much gene expression is there for the particular cancer patient. So you have to remember, if you want to measure the gene expression, so you should also have a sample of healthy human. Okay, if only if you have a healthy RNA, then only you can try this hybridization protocol with the diseased RNA. Okay, this is something about the gene expression, and whatever gene expression you're going to estimate, in NCBI, we can able to find this data, the expressed information, express gene information. You can find data in the form of ESTs, express sequence tags, and there is also one more kind of a data in the form of data set. This you can find in the form of a data set, that is GEO, gene expression omnibus. Like if you have taken 160 human samples and you cluster together in the form of a single data set, so you can find the information of data sets in GEO. So GEO is, of course, you can also find even the hereditary informations. Like even if you want to check whether a cancer is going to be heritable from the parents or not. Okay, if any parents have a hereditary kind of a cancer. So for all of these things, it can help you to understand about usage of these databases, EST and GUO present in NCBI. Okay, now how a hybridization chamber looks like. This hybridization chamber, you have to place a microarray inside, and you have to maintain humidity, temperature, and formamide for binding for hybridization, for hybridizing one RNA with another RNA. You know, even this hybridization chamber is used for hybridized DNA and RNA, or RNA, single RNA versus single RNA. So if hybridization happen with DNA and RNA, it is a single stranded DNA with RNA. But in our case, we generally use RNA of disease and RNA of the healthy, and then you finally estimate the analysis from the colored spots. I told you in normal cells, if it is green, if the equal expressions are yellow, in pathological cells, you get like red color. So whatever I explained, you can find this information. So microarrays are also, if you see the steps, these are the steps we can find. So where we can find bioinformatics analysis. So in image analysis and acquisition and normalization of the data, okay, and clustering of data, discrimination, testing, estimation, in all this application you find bioinformatics applications. And of course, uh, this information is stored in the form of data sets or data in various databases. In NCB itself, we have majorly two databases, but also we have other public and private domain databases for the gene expression data. So gene expression assays. So these are the assays. Okay. So these are the modified assays. So you remember in next generation sequencing, I was explaining you about uh, Illumina sequencing. So if even if you take Illumina sequencing, the Illumina sequencing is nothing but a modified Sanger method of sequencing. Okay, you are using an enzymatic method, but you are using an advanced, that is a modified kind of a Sanger sequencing method. Similarly, in microarrays, you can modify it to get a kind of a best gene expressions analysis. So one such a modification is the SAGE, serial analysis of gene expression. So you can also try with a few more kind of assays. These are the assays for measuring the gene expression. So what is the application? Gene expression profiling, estimating to a DNA copy number, molecular diagnosis, drug development, measuring mRNA decay rates, identifying protein binding sites, of course, in pharmacogenomics. So even if you want to identify protein binding sites, one has to use the microarrays. Gene expression by SAGE. So this is one such protocol showed. Okay, instead of a single tagging with fluorophore, you have a ditags tagged with the molecule. So you get a kind of a best kind of a gene expression with SAGE. This is an extract from Primrose and Wyman. Okay, genomics by Primrose and Wyman. Then the types of DNA arrays for expression analysis. You know, if you see the microarrays, I told you we have DNA microarrays as well as protein microarrays, isn't it? So if you see the protein microarrays are generally carried out using this material, spotted nylon microarrays. Okay, but here it's a macro. They showed you microarrays, but we have also nylon microarrays for protein-protein for checking the protein-protein interactions. So in this table, they mentioned like the difference between spotted nylon macroarrays, spotted glass microarrays, and Affymetric gene chips. So many try the DNA expression, that is DNA expression, using spotted glass microarrays. Okay, just because of cheaper, but now you can also find this Affymetric gene chips discovery gives a wide range of applications. So many are also trying this. This is a old technique. This is a kind of a new technique. Okay, so many parameters are mentioned. Of course, this is present in Primrose and Feynman textbook. So spotted DNA arrays, oligonucleotide chips. These are the types. Okay, from the Primrose and Feynman. So that is spotted DNA array. you're going to use a glass chip. Okay, so where you are going to feed the DNA and you go with the how you whatever I mentioned. First, you have to take an RNA, go with the conversion of cDNA by reverse transcription. So tag with the fluorophore and you do all this, okay? And in oligonucleotide, instead of taking that RNA to cDNA conversion, okay, here we take a small nucleotide. You're not going to take a large nucleotide, but a oligomer is a shorter length in size. That we take and we use and we go for we use similar kind of microarray steps for determining the gene expression analysis. So finally, protein microarray. So if you're using glass microarrays for DNA expression, that is gene expression, for protein-protein interactions, you can go with protein microarray where you use nylon as the material. So here in this example, they have mentioned like reactive epoxide surface chemistry binds proteins covalently in several different ways, and therefore a high number of protein molecules can be captured on the surface for various binding reactions. If you want to estimate the protein binding sites, okay, so protein microarrays are generally used. Okay, so you can find in this region, okay, super epoxy region is attaching, okay, is attaching a protein to the probe, nylon probe. So that kind of an attachment can be amine based, or even it can be hydroxyl based, or it can also be a thiol based. So these are the functional groups which is attaching our protein to the probe, nylon probe, okay, through the super epoxy resin, through the super epoxy resin. So this is how a protein can be interacted. You have heard about sandwich ELISA. So you have an antigen. So from the antigen, you find the primary antibody. Okay, and the primary antibody is generally linked with secondary antibody. If this is a primary antibody, the primary antibody is linked with secondary antibody, and secondary antibody is linked with a substrate. And if you attach a fluoro and if you find an expression with the substrate, you find a different color, yellow to blue color in sandwich assay, ELISA. So the protein-protein interaction, that is attaching of primary antibody to the secondary antibody. So this kind of attachment, conjugation, we can able to find using the protein microarrays. Through the gene expression, through the protein expression, one can able to know whether a primary antibody is binding with the secondary antibody or not. So protein-protein interactions are best formed from that. The final concept in microarrays is to understand the clustering technique. If you have numerous data, if you can able to link, that is if you have gene expression data, okay, and if the genes, or if you go for a protein-protein interaction, if you find proteins which are closely linked with each other, so this it can agglomeratively can cluster. And hierarchical clustering will be like this. You can be protein 1, protein 2, protein 3, protein 4. Protein 1, protein 2 is linked with each other, and protein 2 and protein 3 and 4 is linked with each other, but protein 1 and 2 can also link with the 3 and 4 to the hierarchical clustering. And nearest neighbor, okay, so which is close out of all these proteins. So of course, if you see in this cluster, and if you see in this cluster, okay, this protein is close to this. Okay, so this is the nearest neighbor single linkage clustering. If you want to link completely, that is, this is the maximum span. This is the maximum span we can find. This is a complete linkage clustering. And likewise, average linkage clustering. Most of the length, okay, of the clustering is remaining same. Most of the clustering has a kind of an equal length. Here we have some variation lengths, but if you have nearly thousands of data, you can find, so and you can take just that alone as average linkage clustering. K means clustering. In K means clustering, if you consider as K equals 1, okay, this line is K equals 1, whether you can able to see, so think like this is a protein 1, this is a protein 2, this is a protein 3, protein 4, whether it can, and similarly, this is another class of protein, this is protein 1, protein 2, protein 3, and protein 4. So if you use k equals 1, can you tell 1 is linked with 2 or 1 is linked with 3 or 4? You can't. Okay, if you just ignore this. So from k equals 1, you can't able to know which protein is linked with what. If you take k equals 2, if you take k equals to that this line, okay, so from this it is visible now that 1 is linked with 2 and 3 is linked with 4. Similarly here 1 is linked with 2, but I don't know what's that, how the 3 and 4 is linked, because in this line, okay, this is not visible, this is hidden, the top one is hidden, so you can't able to find here. But here I can tell 1 2 is linked, 3 4 is linked. So one has to go for another k equals 3, something like this. K equals 3. So drop a straight line. You find k equals 3. Now you can check. Even 1, 2 is linked and 3, 4 is linked, and 1, 2, 3, 4 can also be linked. Here 1, 2 is linked. 3 is linked with 1, 2. But still I don't know what is about protein 4. Then you have to take one more line. That is k equals 4. In k equals 4, now it's clear. Okay, but for k equals 3 is sufficient for this protein family, protein cluster, but for this protein cluster you need k equals 4. So this is what k means cluster, how many k you need. Okay, so this is the end of microarrays. Okay, so I've told you even what is microarray and what for it is used, and what are the types of microarray, DNA and protein microarray, and how what is a clustering technique, and what are the materials of construction of uh, microarrays. And then we have initially seen what is system biology approach, and we have seen various protein-protein interaction databases, DNA protein, carbohydrate protein interaction databases. Then we have gone for synthetic biology and applications of synthetic biology. Any doubts you can ask me at present. You have any doubts? If no doubts, thank you very much for the interaction and patience. Have a great day to you all.